So the previous class we have completed human health and diseases, and today we'll start with the new chapter, that is microbes in human welfare. Microbes in human welfare. So before this microbes in human welfare, we are having the chapter. The chapter is animal husbandry. And this chapter is deleted. Okay, so this chapter is deleted. So therefore, you need not focus on this chapter, and rather you will get into microbes and human welfare. Okay, so straight away we get into microbes and human welfare. See, what are microbes? Microbes are nothing but as a microorganisms. The microorganisms which cannot be seen with our naked eye, it can be seen only under a microscope. Particularly, it can be seen only under the transmission electron microscope or scanning transmission, still we used to call it. Scanning transmission electron microscope. So, the organisms which cannot be seen with our naked eye, and it can be seen only under the microscope, microscopic examination. There is such type of organisms, we are calling it as microorganisms. What are those microorganisms? So, the microorganisms can be from plant cell it starts from virus, bacteria, and fungi. We'll take these four examples. Microorganisms. The primitive fungi such as the yeast, Saccharomyces cerevisiae. So the yeast mutate, and this cannot be seen under the naked eye. It can be seen only under the compound microscope, like virus, bacteria, algae, and fungi. When you take this virus, the virus is the major artist causing infections. Causing the infections, or otherwise causing the disease. So therefore, we can consider this virus as a pathogen. What is a pathogen? The organism which causes the infection, which causes the disease. That means it is hazardous. Virus is considered to be hazardous. It is an hazard. So therefore, we need not study under the human welfare. Since it is going to be hazardous, it is going to cause an health ailment. So we cannot study this virus under the human welfare. Rather, the virus will be considered as a pathogen. So therefore, we have studied about the virus in human health and diseases in the previous chapter. So now we are not going to see about the virus here. Now coming to the next microbe, the microorganism will be the bacteria. When you take out this bacteria has both beneficial as well as pathogenic character. As well as what? Pathogenic character. It has both the beneficial as well as pathogenic. Pathogenic means disease causing. Pathogenic means disease causing character. Whereas beneficial means what? Which is having human welfare. Which has human welfare. Say for example, you can take in the production of antibiotic, in the fermentation process, in the production of vaccine, in the production of flavoring agents, in the production of fermentation antibiotics, vaccines, in the production of enzymes, So industrial production, what we are calling? We are calling it as economically important. Economically important. It is what? economically important in nature. It is economically important, and this shows it is human. It has the human role in the human welfare. So we have to study about bacteria. Next, what we have is algae. See, this algae is a very good organism. Why it is considered as a good organism is this algae do not cause any pathogenic effect, do not have any pathogenic effect, and similarly 99% of algae will not have what pathogenic effect, and similarly they are eco-friendly in nature. They are eco-friendly in nature. That is the importance. Other than that, the algae has got no important applications. Sometimes in agar production, for the culture, media growth, microorganism growth, the highest classes you will be studying. Some industrial, pharmaceutical productions there, some cheese industries where it can be used. Eco-friendly is main role, and some industrial production also will be there. So there is no pathogenicity, pathogenic effect we will not notice in algae as far as algae is concerned. And the next one what we have is fungi. So fungi when you take as like bacteria, it has more pathogenic effect, and similarly in the industrial production that is human welfare. For human welfare also we can use. For human welfare also we can use. So these are the various microbes here we are going to study. So we are going to focus only on bacteria. The other one is the fungal organisms. Fungal organisms. So in this chapter we are going to study only about those two organisms, that is bacteria and fungi, and its economic importance in the production of various substances. Like first we go for the first one is you take the microbes under household production, the products or a manufacturer. What are the microbes that we are going to use? The first one is second one. Okay, then third. The first one is in the bacteria, the organisms we can call it as Lactobacillus lactis, or otherwise it can be called as lactic acid bacteria. This is LAB, lactic acid bacteria. So Lactobacillus lactis. Second one, in the fermentation process. In the fermentation process. And third one is in the production of cheese. In the production of cheese. Cheese industry. Cheese production. In the cheese production. Next, microbes. Under microbes under the industrial production. So microbes under the industrial production we have. First one is about the antibiotics. Second one is enzymes. Then the third one is cyclosporin A. Then fourth one is production of cyclosporin A and stat. Very important. Complex metabolism. Cyclosporin A, enzymes and antibiotics. So we are going to see only these topics today. Okay, first one is Lactobacillus lactis. What is this Lactobacillus lactis? This Lactobacillus lactis is a bacteria which is present in the milk. And this Lactobacillus lactis is very essential for converting the milk into curd. It is also a kind of fermentation process. What is fermentation? Fermentation is an anaerobic process. What is anaerobic process? Any process or any mechanism or metabolic activity which occurs in the absence of oxygen, then we call it as what? Anaerobic process. Anaerobic process is a process that occurs in the absence of, that is actually we are in need of glucose, and the glucose will be metabolized to give rise to the formation of a pyruvate, two molecules of 3 carbon compound in the lower class of table. In the pyruvate, in the absence of oxygen, will get decomposed, giving rise to the formation of lactic acid. Epepella anaerobic process nadakudho, then the carbon dioxide gases will be getting liberated. You understand what I am telling? And sometimes ethanol will also be produced. So in the carbon dioxide manufactured panni varakudiyadhu will be widely used as the dry ice. So lactobacillus lactis, we'll see now. And what are the points that we have to write? In lactobacillus lactis, we'll see. Lactic acid bacteria. See, this lactobacillus lactis helps in conversion of milk into curd. That is, this curd will be having this rod-shaped bacteria. Microscope la pata, then we will not consider curd only. So n number of lactobacillus lactis bacteria will be present in the curd. Okay. Now, actually, this curd, meager amount of the curd, when it is getting inoculated. Inoculation. What is that inoculation? Inoculation, transferring the microorganism. What is a microorganism? Lactic acid bacteria. That is lactobacillus lactis, which is present in the curd, is getting transferred. That is, it is getting inoculated into the milk. Then what will happen? The milk will get fermented, will get converted into a curd. So first point, it helps in conversion of milk into curd. First. Second thing is, this bacteria, LPAB bacteria, helps in partial digestion of milk protein. What is the milk protein called? Casein. Milk protein is called as what? Casein. So partial digestion of milk protein, which is called as casein. And the next point is this LAB, lactic acid bacteria, this is LAB, helps in the production of or increases the content of vitamin B12 in the body. So increase the concentration of vitamin, very important topic, vitamin B12 in the body, in the blood. So when vitamin B12 is present, then obviously it results in preventing or prevention of pernicious anemia. Which occurs due to the depletion of RBCs, results in anemia. So it prevents from the pernicious anemia. And similarly, this lactic acid bacteria concentration in the stomach prevents other pathogenic microorganisms to be present in the body, to be present in the body. to be present in the body. Okay, so this is about what? Lactobacillus lactis. So what is the first point? So if lactic acid bacteria is going to be present in the body or present in the stomach, that makes or renders other pathogenic microorganisms inactive. So the pathogenic microorganism cannot exhibit its metabolic activities inside the stomach provided if this lactic acid bacteria is going to be present. Can you understand? So what is the first point? It helps in the conversion of milk into curd. So curd, when it is being inoculated into the milk, it undergoes a fermentation, giving rise to the formation of a curd. So then lactic acid bacteria, partial digestion of the eggs and partial digestion of milk protein, which we are calling as casein. The lactic acid bacteria increases the concentration of vitamin B12 in the body or in the blood. So vitamin B12 prevents from the pernicious anemia. The lactic acid bacteria concentration in the stomach prevents other pathogenic microorganisms to be present in the body or in the stomach. So this is the main role of lactobacillus lactis. So this lactobacillus lactis can be produced in the house itself. You buy from when you get a small inoculum of the curd, when you get a small amount of the curd that can be inoculated into that of a milk. So then the milk will undergo some fermentation, giving rise to the formation of a curd. So here only 5 to 6 organisms which are going to be inoculate. Here you will be getting 50 to 60 organisms to be formed. So proliferation, multiplication. So this can be done under the room temperature. Under the room temperature, normal condition. So we need not refrigerate, we need not have any in vitro conditions and all those things. In vitro or in vivo condition, nothing is needed. Only the room temperature will be more than enough for the organisms to proliferate or otherwise to multiply. So that is why we are calling the microbes which can be manufactured under the household product for the production of the household goods. Can you understand now? So this topic is over. Now the fermentation process. In the fermentation process there are lots of organisms. Baking industry and brewing industry in the body. Isn't it? So it can be widely the fermentation industry அப்படிங்கறது ferment ஆன ஒரு பெரிய tank vessel. So the fermentation baking industry. The other one is brewing industry. In the fermentation, very important organism is Saccharomyces cerevisiae. So this is nothing but unicellular yeast. Unicellular fungi is a prokaryotic nature could be a fungi, unicellular fungi. So which is why we use fermentation in the industrial method in a natural homemade baking industries, industry level brewing industries, it can be widely used. Normally as well as Saccharomyces cerevisiae. For baking industries, what is the principle? What is the process? So we are taking the flour or otherwise dough. So this becomes how what's happening to this flour. So it will be getting puffed or it gets softened, isn't it? So this is getting softened due to the liberation of carbon dioxide. I told you in the fermentation process, it results in the liberation of carbon dioxide, production of carbon dioxide. So the dough gets puffed or otherwise it can be called as softened with the liberation of what? With the liberation of carbon dioxide. So therefore you can see the dots. Not only in the baking, bread, not only for the bread manufacturing, not only in the bread, but also for the rice flour to make idlis and dosa. So in idli and dosa, you can see the holes, isn't it? The pores or the rice holes. The holes or the pores indicates the production or liberation of carbon dioxide. Liberation of carbon dioxide. We are calling it as the liberation of water, liberation of carbon dioxide in the vapors. And similarly, next example is toddy. It's very important one mark. So 100 and more than 100% this question will be asked as well as a NEET as well as a board examination. What is this toddy? Is produced by the fermentation of palm sap. Palm tree, you know, and the palm tree will put a fruit and the sap, exudate sap. When it is fermented, we will be getting toddy. It is nothing but an alcoholic beverage. It is otherwise called as the alcoholic beverage. And similarly, this Saccharomyces cerevisiae can also be used in the fermentation of fishes. Dry fish will get fermentation of fish. Then bamboo shoot. This is also very important. The bamboo shoot is widely used in the manufacturing of pickles. The manufacture of pickles. So production of pickles or preparation of pickles we can widely use. That is fermentation of fishes ferment பண்றதுக்கு pickles we can use. And similarly in the soya bean. This is the fermentation. இல்லையா? So அப்ப ஞாபகம் வச்சுக்கிறது the liberation of carbon dioxide results in due to the puffness that can be confirmed by the puffness. So fermentation process attenawe liberation of carbon dioxide, production of carbon dioxide. So not only we can use flour or jaw, and also we can manufacture toddy. Not only bread, idli and dosa, apart from that toddy. Toddy is nothing but what? It is a fermented alcoholic beverage which is obtained from the sap of the plant. What is sap? Sap அப்படிங்கிறது it is a fluid content or otherwise it is a kind of a nutritive edible part of the fruit which is present within the plant, and when it is being fermented it gives rise to the formation of toddy, the fermented product called as toddy. The other one is fermentation of fishes, bamboo shoots, and similarly soya beans. Bamboo shoots for the preparation of pickles we can widely use. Okay, so this is also very important and you have to study. The next one is brewing industry, isn't it? The microbes, the brewing industry we are telling. Similarly, Saccharomyces cerevisiae in the manufacture of wine and tea, and similarly whiskey, brandy and all can be manufactured, which involves the fermenter. Fermenter we are telling the malted cereals and fruit juices and brewing industries there, which helps in the production of alcoholic beverages. Same Saccharomyces cerevisiae will be widely used. Malted cereals. What is this malted cereals? Sometimes it can be molasses, sugar molasses. So malted cereals on further fermentation, it gives rise to the formation of this ethanol and carbon dioxide. This carbon dioxide will be widely used as what? Will be used as the dry ice. Okay, ethanol. So for this brewing industry, we will be using the fermentor. Fermentor is nothing but it is a large vessel where these microorganisms and the raw materials will be getting fed into that of a vessel, and under the optimum conditions of temperature and pressure, the production of this alcoholic substances, beverages, can happen on a large scale, on an industrial scale. So the fermenter tank, what is a fermenter tank? Fermenter tank it is a large vessel. It is a large vessel. It is a large vessel, and similarly, this fermenter will be has to be fed with the raw materials. The the raw materials plus microbes, and it should have anaerobic temperature, anaerobic condition. Anaerobic condition, what is the anaerobic condition? Absence of carbon dioxide, absence of oxygen, anaerobic condition. So the raw materials and the microbes has to be fed into the complete ferment or tank, isn't it? So the large scale or the large scale alcoholic beverages can be manufactured. Brandy, whiskey. So some of the most examples are there. No? Brandy, whiskey, wine, beer, and all we can obtain, isn't it? So this alcoholic beverages, wine and beer, direct ferment without distillation, without purification, without distillation, we are calling it as wine, beer. Suppose with distillation, if we get with the distillation, if we get, then we are calling it as whiskey, or otherwise brandy, or otherwise it is called as brandy. Can you understand? Whiskey or otherwise we are calling it as brandy. So baking industry, the other one is brewing industry. Next one is cheese. So fermentation. Next what we have is the cheese, isn't it? So we go to the cheese. So the next one is cheese. Cheese land, we have two types. One is Swiss cheese. The other one is Roquefort cheese. Roquefort cheese. This Swiss cheese is a bacterial cheese. It is a bacterial cheese. Roquefort cheese is a fungal cheese. It is a fungal cheese. Okay. Now, very important one more. The Swiss cheese is obtained from bacteria coli. What is the bacteria called? Propionibacterium shermani. And the other one is, the next one what we have is Roquefort cheese. This Roquefort cheese, when you take this Roquefort cheese, is obtained from Penicillium Roquefort. Penicillium Roquefort. So Roquefort cheese is what? Penicillium Roquefort. So there are two Swiss cheese. The other one what we have is the Roquefort cheese. So Swiss cheese Propionibacterium shermani, and Roquefort cheese Penicillium, it can be widely obtained. So here large scale it can be produced with the production of carbon dioxide. So in case of Swiss cheese, you can see the liberation here also, the liberation of carbon dioxide resulting in large holes. So you can see large holes on the cheese. Propionibacterium shermani, Swiss cheese, you can see the large holes to be present, that is occurring due to the liberation of carbon dioxide. The next one is Roquefort cheese, that is obtained from what? Penicillium. It is ripened by growing a specific fungus. So this Roquefort fungi cheese is mainly essential for flavoring. It is widely used as the flavoring agent or flavoring substance. It is widely used as the flavoring agent. So Swiss cheese, the other one is Roquefort cheese. So in the cheese industries, it is widely used. So lactose, we have studied fermentation, the baking industries and brewing acidic we have studied. The other one is called as what? Cheese industries that we are widely using. The next one what we are having is antibiotics. Antibiotic அப்படிங்கிறது what is that? Antibiotic அப்படிங்கிறது chemistry-லையும் I have explained you, biology-லையும் I have explained in the previous classes. Antibiotic is a substance, chemical substance which is given, which is extracted from chemically or otherwise from certain microorganism in an inactive state and which gives the increase the production of antibodies or gives resistance mechanism to the disease causing pathogen. There are disease specific antibiotics are there. These antibiotics are widely given for various diseases, bacterial disease, viral disease, say for example diphtheria. So for that antibiotic DPT in the form of vaccine is given, diphtheria, pertussis, tetanus. Similarly, measles, MM1 vaccine in the form of antibiotic. Actually, it's an antibiotic which will be given in the form of a vaccine. It's an immunization for the measles, mumps, rubella, against rubella, mumps, and then against measles it will be given. And similarly, for the bacterial infection, very dreadful sexually transmitted disease, there is syphilis. Syphilis we are doing. This syphilis when you take, this syphilis is an infection, bacterial infection which is caused by Trypanosoma palladium, which is caused by sexually transmitted disease, which is caused by the bacteria, And similarly for the whooping cough, dry cough, continuous whooping cough. So, n number of antibiotics have been discovered so far. From bacteria as well as from the fungi. So various antibiotic, tetracycline is there, penicillin is there, cephalosporin, these are the antibiotics are there. The exact examples are what I am writing. Cephalosporin, penicillin against syphilis can be given. Cephalosporin is there, penicillin is there, tetracycline is there, So the cephalosporin it is a powerful antibiotic. So Staphylococcus aureus, methicillin is also important. Neat exam methicillin resistant Staphylococcus aureus. What is this methicillin resistant Staphylococcus aureus? You can write it as MR Staphylococcus aureus. AUREUS. Methicillin resistant Staphylococcus aureus, which can be written as MRSA. It is a very, very powerful antibiotic. Which is a powerful antibiotic. MRSA, which is a powerful antibiotic, strongest antibiotic, and it can be used as the superbugs also. It is widely used as what? Superbugs. It is widely used as superbugs. Okay, it is widely used as superbugs. What is superbug? Staphylococcus MRSA can be widely used as superbugs. MRSA. What is this MRSA? I have written there methicillin resistant bacteria is Staphylococcus aureus, and it is widely used as the superbug. Here the name component of MRSA is registered because it is specific and inactivates specific to, it It is specific to bacterial strains. It is specific only to the bacterial strains. It is specific only to the bacterial strains. Okay. Apart from that, industrially, the fungi called Aspergillus niger. It is a fungi. Aspergillus niger, on a large scale, production of Aspergillus niger helps in the manufacturing of citric acid. Okay. Then bacteria called as Acidobacter aceti helps in the manufacture of acetic acid. Then Saccharomyces cerevisiae. Saccharomyces cerevisiae. Saccharomyces cerevisiae. Learn the spelling. Saccharomyces cerevisiae can be widely used in the production of alcohol, particularly ethanol. So Saccharomyces cerevisiae. And the next one is Acidobacter. Clostridium botulism. What is the next example? Clostridium. The next example is Clostridium botulism. Clostridium botulism is widely used in the production of butyric acid. So, what are the organisms? Aspergillus niger, citric acid. Acidobacter aceti, acetic acid. Saccharomyces cerevisiae, ethanol. Clostridium botulism. So, Clostridium botulism is butyric acid. Previously we have studied lactobacillus. This lactobacillus helps in the fermentation process. So you can see the organism also, and I have shown the pictures that you can see in the pictures. So Aspergillus niger, citric acid, Acidobacter aceti, acetic acid, Saccharomyces cerevisiae, ethanol, Clostridium botulism, butyric acid, Lactobacillus, lactis in the fermentation process. Again, Aspergillus niger is a fungi. Aspergillus niger is a fungi which helps in the production of citric acid. Acidobacter aceti helps in the preparation of acetic acid. Acetic acid, vinegar. As a preservative it can be used. Then Saccharomyces cerevisiae, ethanol, fermented product. Ethanol will be there, which is used in the brewing industries. Then as a beverage. Then Clostridium botulism, butyric acid. Butyric acid is also a kind of a preservative. The lactobacillus helps in the fermentation process. So these are some of the industrial uses. So antibiotics I have given you. So antibiotics MRSA, superbug. What is superbug? Then what are the different organisms widely used in the industrial production? And next one what we have is in the enzymes. So microbes, industrial production there, very important is in the enzyme, the production of enzyme. We'll see now. The production of what? The production of enzymes. In the production of enzymes. Enzymes are a biocatalyst which brings about various biochemical reactions happening in the body. So enzymes. Enzymes are here we have studied. So few examples we see. First one is lipase. Second one is protease. And pectinase. Then streptokinase. Very important. This also you can expect in your board examination. What is this streptokinase? This streptokinase is an enzyme which is obtained from Streptococcus bacteria, and this streptokinase is considered to be a clot buster. It is considered as what? It is a clot buster, where the clots will get burst when you use the streptokinase in the blood vessels. When there is a clot, and then this streptokinase will go and pierce this clot. So as a result, it prevents from the angina pectoris. Angina, severe pain. Angina pectoral region, pectoris. Angina pectoris. Silent ischemia, mild thought attack, severe attack. Okay, silent attack, ischemia. So from all these things it will be giving the protection, isn't it? So silent ischemia, angina we are calling, and then for even the myocardial infarction, even it prevents from the, ultimately it prevents from the myocardial infarction. Prevents from myocardial infarction. Clot removing. So then this protease and pectinase is used as juice clarificating agent. It is considered as what? Juice clarificating agent, because the pomegranate juice you would have seen the turbid will be getting formed after putting it in the mixing. After filtering the seeds, some OS particles will be present. When it is being filtered, and we are taking it in a container and keep it aside for some time, and you can see the turbid to be formed at the base of the container. So when there is a turbid is going to be present at the base of the container, and only when the suspended particles are fluid, the liquid part alone is going to be present at the surface, that is a heterogeneous mixture that will not be comfortable to drink also. So what has to be added? Either pectinase. The pectinase will depectin the substance. So pectin is a cell-wall substances that will be present on the surface of the seeds and as well as on the fruit layer. So that has to be degraded. That has to be diluted. So it can be diluted by using an enzyme that is called as pectinase. What is so then the turbid formation will be getting clear, and there will not be any side effect because it is not an artificial substance and it has been synthesized only from the plants and some microorganisms like bacteria. Protease, it helps in the digestion of protein, breakdown of a protein. So protein has to be broken down. Complex protein should not be present, so that gives some discomfort to the intestine and discomfort to the body in order to secrete the enzymes which is essential for the breakdown of a protein. So when the protease is being taken, that results in masticating the protein molecules. So breaking down the macro substances into micro substances. So the protease and the pectinase is widely used as a juice clarifying. The Tropicana manufacture Tropicana juice, you cannot see the turbid. Nowadays they are manufacturing, isn't it? The pulp of fruits. Sometimes they will be using this protease and even pectinase. So then it will not be getting restored at the base. It will not be getting supplied. Turbid precipitate matter will not be formed at the base of the container. So what is an enzyme? The enzyme is called as what? Protease. The other one is called as pectinase. The next one is the lipase. The lipase is widely used to degrade the oil stains present in the clothes. So lipase degrade the oil stains which is present in the clothes. Protease and pectinase, juice clarifying agent. Streptokinase is widely used as the clot buster. So it prevents from angina pectoris, silent ischemia and myocardial infarction. Okay, so this is about what? Enzymes. This is about what? Enzymes. The next one what we have is, next one, cyclosporin A, the other one is statin. Cyclosporin A. Next one is cyclosporin A. Cyclosporin A is very important for normal metabolic activities to be carried out in the liver. So it is obtained from trichoderma. So it is obtained from the organism. The organism is called as what? Trichoderma polysporum. Okay, trichoderma polysporum. Trichoderma actually the liver helps in the metabolism of lipids. Metabolism of the lipids. When the lipids are not getting properly metabolized due to the malfunctioning of the liver, then automatically in the blood vessels, there will be deposition of the lipids. This is called as what? Lipid deposition. It results in lipid deposition. The lipid deposition is resulting in atherosclerosis. So there will be a deposition. What is getting deposited? The lipids will be getting deposited, and as a result, it gives rise to the formation of atherosclerosis. So cholesterol deposition. So blood vessels will be getting locked. So as a result of atherosclerosis, the blood vessels will be getting blocked. The blood vessels blocked. Isn't it? Because of cholesterol deposition. Due to the cholesterol deposition. See, this lipid deposition can otherwise be called as a plaque. It is a plaque. Plaque is a deposit. MAC molecular substance form பண்ணிடும். So this gives the hindrance for the blood flow. RBC blood flow hinder பண்ணக்கூடியதுனா இந்த plaque. Or otherwise the lipid or the cholesterol deposition. So when the cyclosporin is getting taken and this clears, what it clears? The fatty or the lipid depositions on the blood vessels. So it enables the liver cells to get here to perform its metabolic activities in a proper way. Okay. The next one, what is similarly, this cyclosporin A is also used as an immunosuppressive agent in organ transplant. It is widely used as second point, it is widely used as immunosuppressive agent. எப்படிது அதுதான் என்ன சொல்றோம்னா host versus graft. Host versus graft. And also it helps in the production of MHC. This is called as immunology major histocompatibility complex. Histocompatibility complex. Production of major histocompatibility complex. The other one is called as major histocompatibility complex, and the production of HLA. What is this HLA called? Human leukocyte antigen. So idellame immunosuppressive agent. What is that? See, if I am a host, so I am a host. இல்லையா? இப்போ நான் host ஆகிறேன். எனக்கு graft or otherwise organ transplant. Graft என்ன அர்த்தம்? இப்போ say for example skin burn ஆயிடுச்சு. So என்னோட கையில skin எடுத்து அந்த இடத்துல transplant பண்றாங்க. வேற ஒரு ஆர்கனிசத்துல இருந்து skin எடுத்து அந்த நம்ம உடம்புல இருக்கக்கூடிய ஸ்கின் எடுத்து they will do the plastic surgery, or otherwise வேற ஒரு ஆர்கானிசம் இல்லாட்டி சேம் பிளட் குரூப் இருக்கக்கூடிய நம்மளுடைய ஃபேமிலியில் இருக்கக்கூடிய ஸ்கின் மிஸ்மேட்சிங் ஆகாமல் இருக்கிறதுக்காக, கிராஃப்ட் ரிஜெக்ஷன் ஆகாமல் இருக்கிறதுக்காக they will do the plastic surgery. அப்படி பண்ணும்போது நம்ம உடம்புல இருக்கக்கூடிய immune system should be suppressive. It should be suppressed. It should not be immunocompetent இருக்கக்கூடாது. Immune system should be suppressed in order to not involve in graft rejection. இப்போ graft என்ன தெரிஞ்சிருச்சு, வேற ஒரு ஆர்கானிசத்துல இருந்து ஸ்கின் எடுத்து என்னோட சர்ஃபேஸ் ஆஃப் த ஸ்கின் எங்க பர்ன் ஆயிருக்கோ, எங்க பிளாஸ்டிக் சர்ஜரி பண்ணி அழகுபடுத்தணுமோ அந்த இடத்துக்கு வைக்கக்கூடிய grafting. It is a surgery, plastic surgery. இந்த பர்ன்ல வரதுனால எடுத்து ட்ரான்ஸ்ஃபர் பண்ண முடியாது. அப்படி பண்ணும்போது இம்யூன் சிஸ்டம் எப்படி இருக்கணும், சப்ரஸ் ஆகி இருக்கணும். அப்ப இம்யூன் கம்பீட்டண்டா இருக்கக்கூடாது. It should be compromised or otherwise it should be suppressed, so then the graft will not be rejected. Clear? அது 1 or 2 days இல்ல அந்த பர்சனோட இம்யூன் சிஸ்டத்தை பொறுத்து 48 hours, 24 hours, 72 hours, so something 18 hours, something like that, depending upon the ability of the person பொறுத்து, அவருடைய லைஃப் ஸ்டைலை பொறுத்து, பாடி ஃபீச்சர்ஸ் மெக்கானிசத்தை பொறுத்து the doctor will give this medicine. இவ்வளோ நேரத்துக்கு இம்யூன் வந்து சப்ரஸ்ட் கண்டிஷன்ஸ்ல it should be present. That is called as the graft. Host versus graft. Sometimes organ transplantation. So இப்போ ஒரு கிட்னியை ஒரு பர்சனுக்கு நம்ம ட்ரான்ஸ்ஃபர் பண்றோம்னு வெச்சுக்கோங்க, ஆர்கன் டிரான்ஸ்பிளான்டேஷன். அப்ப சேம் பிளட் குரூப் இருக்கணும். சம்டைம்ஸ் அதுல ஏதாவது லிபிட் ப்ரொஃபைல் அதிகமா இருக்கு, ஆல்கஹாலிக் டெபாசிஷன்ஸ் அதிகமா இருக்கு அப்படின்னா அதே ஒரு ஹெல்தி பர்சனுக்கு கொடுக்கும்போது though the blood groups are similar, that results in rejection. That results in what? Cross reactions அப்படின்னு சொல்றோம். That we are calling it as a rejection. Agglutination. நம்ம blood எல்லாமே என்ன ஆக ஆரம்பிச்சிடும், rejection வந்தாவே blood will start to clot. It disable itself in order to carry out the oxygen. So அப்ப blood எல்லாமே agglutination mechanism குள்ள போகும்போது blood clot ஆச்சுனா it becomes lethal to the individual. அப்ப அந்த இடத்துல rejection should not take place. So again the immune system has to be suppressed. That can be done by using major histocompatibility complex and human lymphocytic antigen ங்கிறது நார்மலாவே நம்மளுடைய இம்யூன் செல்ஸ்ல இருந்து இம்யூன் ஆர்கன்ஸ் வந்து ப்ரொடியூஸ் ஆகக்கூடியத ட்ரிகர் பண்ணக்கூடிய ட்ரக் இஸ் சைக்ளோஸ்போரின். சோ இம்யூன் சிஸ்டம் சப்ரஸ் ஆயிடுச்சு, இந்த ரிஜெக்ஷன் எல்லாம் நடக்கல, போஸ்ட் வெர்சஸ் கிராஃப்ட் சக்சஸ் ஆயிடுச்சு அப்படின உடனே the MHC injection will starts to secrete. Why? Major histocompatibility na mismatching, histona tissue. So tissue rejection எதுவே நடக்காம அக்செப்ட் பண்றது ஹெல்ப் பண்ணக்கூடிய MHC molecules, major histocompatibility molecule, and similarly human lymphocytic antigens அப்படிங்கிறது which is present in the white blood cells, lymphocytic cells present இருக்கக்கூடியது, that also will prevent the graft to be getting rejected. So major histocompatibility complex human lymphocytic antigens produce பண்றதுக்கு, initiate பண்ண, stimulate பண்றதுக்கு ஹெல்ப் பண்ணக்கூடிய cyclosporin A, immune system suppress பண்றதுக்கு during organ transplantation, during the grafting techniques, the cyclosporin A plays a very very important role, and also it helps in the proper cholesterol or the lipid to undergo the metabolism and prevents from the atherosclerosis, that is a play deposition of the fatty substances on the blood vessels. So this is about what cyclosporin A அப்படினு சொல்றோம். Then the other one is called as the statin அப்படினு சொல்றோம். The statin is also responsible for, so statin, we are calling it as what? Statin, you see now, statin, Monospis purpureus. What is the organism that produces statin? Statin is being produced by statin. So chemical is a substance that is being produced by Monospis purpureus bacteria. And this statin is nothing but it is a cholesterol lowering agent. It is a cholesterol lowering agent. So it helps in normal metabolism, lipids or the cholesterol in the body. So then it prevents, as a result, it prevents from the blockage, hypertension, BP, prevents from hypertension, and similarly from the blockages, and from the fatty deposits. So that is the statin. So statin is produced from monospecific spurious. Then it helps in the lowering of cholesterol level in the body, and it helps in the normal metabolism of lipids and the cholesterol, and it prevents from the blockage. Blockage means blood vessels should not be get blocked by the lipids, which results in myocardial infarction. So blockages, hypertension, and fatty acids. Okay, so these are the various topics which I have taken today, and see to that you are studying properly. Monospecific spurious, MONAS. Monospecific spurious is a bacteria. Okay, what are the things that you have studied? First, the chapter is what? Microbes in human welfare. So microbes in human welfare, first we have studied about milk, epic curd அப்படின்னு சொல்லி படிச்சோம். Lactobacillus lactis அப்படின்னு சொல்லக்கூடிய பாக்டீரியா, இல்ல lactic acid பாக்டீரியா LAB. செகண்ட் fermentation process பத்தி படிச்சோம். சீஸ்னா என்ன, சீஸ் எங்க இருந்து மேனுஃபேக்சர் ஆகுது, அதுக்கு ஹெல்ப் பண்ணக்கூடிய பாக்டீரியா. வாட் இஸ் தட் பாக்டீரியா ஒன்று படிச்சோம், ஃபங்கை ஒன்று படிச்சோம். பாக்டீரியா ப்ரோபியோனி பாக்டீரியம், ஷெர்மானி அப்படின்னு சொல்லக்கூடிய பாக்டீரியா. தென் ஃபங்கை வந்து ராக்கிஃபோர்ட் ஃபங்கை, தட் இஸ் பெனிசிலியம் ராக்கிஃபோர்ட்டில இருந்து நம்ம மேனுஃபேக்சர் பண்றோம். ஃபெர்மெண்டேஷனுக்கு ப்ரூவிங் இண்டஸ்ட்ரி, பேக்கிங் இண்டஸ்ட்ரி, பிரெட் இண்டஸ்ட்ரி, ஆல்கஹால், விஸ்கினா என்ன, பிராண்டினா என்ன, டிஸ்டிலேஷன் ப்ராசஸ் எதுக்கு டிஸ்டில் பண்றோம், பீருக்கு எதுக்கு நம்ம டிஸ்டிலேஷன் பண்றது இல்லை அப்படின்னா நம்ம படிச்சோம். ஃபெர்மெண்டேஷன் டேங்க்ல ஆர்கனிசம் எந்த ஆர்கனிசம் ப்ரூவிங் இண்டஸ்ட்ரீஸ் நம்ம யூஸ் பண்றோம். சாக்ரோமைசிஸ் செரிவேசின்னு சொல்லக்கூடிய யூனிசெல்லுலார் ஃபங்கை வந்து இட் இஸ் வைட்லி யூஸ்ட். அதுக்கப்புறமா ஆன்டிபயோட்டிக்ஸ், நிறைய ஆன்டிபயோட்டிக்ஸ் படிச்சோம். அது ரொம்ப ரொம்ப இம்பார்ட்டன்ட். மெத்திசெலின் அப்படின்னு சொல்லக்கூடிய மெத்திசெலின் ரெசிஸ்டன்ட் ஸ்டெஃபைலோகாக்கஸ் ஆரியஸ் அப்படின்னு சொல்லக்கூடிய ஆர்கனிசம் இஸ் வெரி வெரி இம்பார்ட்டன்ட். அது சூப்பர் பர்கா ஆக்ட் பண்ணுதுன்னு சொல்லக்கூடிய அந்த ஆன்டிபயோட்டிக்ஸ் படிச்சோம். சைஃபலிஸ் அப்படின்ற செக்ஷுவலி டிரான்ஸ்மிட்டட் டிசீஸ்ல வரக்கூடிய அந்த பாக்டீரியல் இன்ஃபெக்ஷன் which can be cured with the help of an antibiotic that is called as penicillin அப்படின்றது படிச்சோம். And then what we have studied, we studied about this cyclosporin A. Next one is what? Statin. Cyclosporin A organism, Trichoderma polysporum. Then statin வந்து Monostochus spurious அப்படின்ற organisms. Okay, till this you study, you complete your classwork, and remaining thing you can continue in the next class. Okay, thank you.